Our DNA is in every cell of our body and codes for everything our body needs, even the way your hair grows. This lesson is on the structure and function of DNA. So, every DNA molecule is composed of chemical units called nucleotides. It also contains a phosphate molecule, a deoxyribose molecule, which is a sugar, and one of four nitrogen bases. So the phosphate and the sugar parts of DNA form a backbone of the DNA. So they're like the sides of the ladder of the DNA molecule. And the nitrogen bases stick out from the sides of the chain and kind of form the rungs of a ladder. This ladder would then be twisted into a double helix shape. Those four different nitrogen bases are adenine, cytosine, thymine, and guanine. The DNA molecule has two strands of nucleotides, and those strands are attached with hydrogen bonds between the nitrogen bases. So, as a result, those two bases bonded together are called base pairs. And there's a set pattern with which nucleotides can bond with other nucleotides, because adenine will only bond with thymine, and cytosine will only bond with guanine. And these are known as complementary pairs. So the DNA consists of two chains of nucleotides with bases paired up with another consisting of a complementary base. So we're able to solve if we were given a DNA strand that said ACT, on one side of the molecule, we would be able to assume that the other side is TGA, CAAT. So if we were given a molecule ATT, we would go TAA, CGA would go GCT, ACT would go TGA, and then A would have a T complementarily bonded onto it. And this is uniform no matter where in the body the DNA is found. So the DNA is coiled into a shape known as a double helix. This helps condense it because it's a very long molecule. And if the DNA from one human cell was stretched out, it would be more than 2 meters long. And this all needs to fit in to one cell. So the genetic material wraps itself around protein spools called histones to condense it. Now, since all of our cells undergo the process of mitosis to grow and repair themselves, we have to make an extra copy of DNA for every process, every time our cell goes through mitosis. And this process is called replication. So to do this, the DNA must untwist, and new nitrogen bases are added onto the existing strand that are free floating in the nucleus and cytoplasm to attach to those DNA halves to produce two new strands. So each strand of DNA that's produced has one old strand and one new strand added onto it. So here we have an image of that DNA untwisting. Those hydrogen bonds are broken between the nucleotide bases, and new nucleotide bases are adding on to produce two new strands of DNA containing one original and one new strand of DNA. So when we look at our genetic code, it's hard to imagine how just four different nitrogen bases can make up such a complicated code. But we can also compare this to things like the alphabet, which is made up of only 26 letters, but has many different combinations of words and sentences in not only English, but other languages. So same with DNA. These four nitrogenous bases provide information needed for all of our cells' life processes. So what our DNA actually codes for is for our body to make proteins that are essential for life processes. Whether this is proteins to make our bones grow, whether this is proteins to make a hormone, or whether this is proteins to make enzymes to help with other chemical reactions in our body. And proteins are made up of individual amino acids. The instructions to make those amino acids are contained on our DNA. And this is actually a group of three of our nucleotides, so a three-letter group makes up the basis of a DNA triplet code. Now, if any of you have taken Biology 30, in Biology 30, we convert our DNA to messenger RNA, mRNA, and we do an mRNA triplet code. But for Science 30, we stick with the DNA. So you'll notice that that chart is different. You can't intermingle them between the Biology 30 data booklet and the Science 30 data booklet. So this is a DNA triplet code. So each of those triplet bases is going to stand for an amino acid. So for example, AAA corresponds to the production of the lysine amino acid. So when you're given a strand of DNA, you're able to correspond that to the amino acids that are going to be produced. Now, all of our proteins start with the DNA triplet ATG, which is the start codon. So if you have anything before then, it doesn't code into amino acid and then protein chains. And then all of our proteins will stop with one of the stop DNA triplet codes, which stops the addition of further nucleotides and releases that protein chain from the ribosome where it's being formed. Our DNA is also redundant. As we can see, there are lots of different DNA codes for some of these amino acids. So for example, we can have arginine coded for in this entire 4 different triplets, as well as these 2 down here. And then we can have gluta or proline, those 4. And for example, leucine, there's 6 different ones here that code for leucine. So we have a lot of different DNA triplets that will end up coding for the same amino acid. So that's our lesson today on the structure and function of DNA.